Okay, today we'll be talking about bacterial growth, nutrition, and differentiation. To start with this lecture, let's go ahead and get into a little bit of vocabulary associated with this. So when we start talking about microbial growth, we're going to be addressing terms associated with growth. We'll also be looking at classification of microorganisms based off of their needs, their essential nutrients, how they make energy, where they obtain electrons from, what are the sources of these products to classify and name microorganisms. Following that, we're going to be looking at how understanding microbial growth ties into pathogenesis. So, how does growth enable microbes to be able to begin an infection within the body? We'll also look at biofilms, a place where microbes are found in nature and can be found in the body as well. Okay. Looking at growing bacteria in a laboratory is a little bit different than finding bacteria naturally in the environment around us. Microbes normally grow. Let's say if we took a sample of soil, microbes grow with hundreds of different species together. But in the laboratory, a lot of times we look at growing microbes, what we call a pure culture. So only one species of microorganisms would be found. The plate you see on the left has different color colonies. We see some with the pigmentation of yellow, orange, pink, white, differential growth patterns associated with these as well. So we see this is a mixed culture, not a pure culture of one microorganism. What we see in the laboratory, in teaching laboratories, most of the time is pure cultures, not necessarily mixed cultures. So we're only looking at E. coli or we're only looking at Micrococcus luteus in one sample itself. The way we grow microorganisms in a lab is by providing them the environment necessary that they need for growth. Sometimes we grow them in liquid media, and sometimes we grow them on solid media, like the plate you see in this image. If we grow them in liquid media, if we have a mixed culture, microorganisms will be mixed throughout. If we grow them on solid media, we're able to kind of differentiate between different types of microorganisms, as seen on the plate in this picture. So we can separate microorganisms on solid media. Okay, if we want to obtain a pure culture, what we can go ahead and do is take a colony that we see from a plate like this, a solid media. So a colony that's not touching any others, we could grab a small piece of that, and that would be our inoculum that we would be using to go ahead and streak out onto a plate. So in order to do this, we have an isolation streak plate, which is what you see here in this image. This starts by doing four different quadrants. Your first quadrant is going to be the most concentrated, and we see that on the plate on the left. Then the second quadrant, which touches the first quadrant, is a little bit less concentrated, and we see that we're starting to see small dots. So those are individual small colonies coming up. Then our third quadrant, we see isolated colonies down to our fourth quadrant, where we have the most dilute sample and the most isolated colonies, meaning they're not touching the colonies, they're not touching the bacteria around them. So we want to remember that just one of these dots that we see on the plate is actually originating from one cell landing in place and then dividing over and over again. So we have millions of cells here to the point where we're actually seeing them on top of the agar surface. Okay. If we want to look at obtaining pure culture and we want to go ahead and count our viable colonies, what we can do here is do a dilution plate. So we can take our sample as a liquid broth and we can dilute it. So we can take 1 ml out of here and add it to here into 9 ml. So that's a 1 to 9 dilution. So it's a 10% concentration that we're adding each time. We do this over and over and over again. What we see is decreasing color in these test tubes. That's supposed to reflect the decreasing concentration of microorganisms. So these are our tenfold dilutions. And what we do with these dilutions is we plate the lower portion of the series. And we plate them and we have a lot of microorganisms here. So many that it's too numerous to count. Over here, a little bit less as we have a tenfold dilution, 254 colonies approximately. again, that's about 10 times, a tenfold dilution less, but still quite a bit too many. Then we can go down here to 23 colonies and then all the way down to 3 colonies, which is almost too few to count on the other end of the spectrum. What we see on the too numerous to count sometimes is what we call confluent growth. So if we were to plate any of these solutions up here and above, we would have entire growth over the entire plate. And that's what we can also call a lawn. What we know from doing this pure culture dilution scheme here is that anything that's found on these plates, once plated and grows, is viable, meaning these are living organisms. So we can reassess the concentration of our original solution based on the viability of the organism. So we're not just counting dead cells here, we're only growing and culturing the living cells. Okay. When we look at growing organisms, we have a couple different types of media that we can grow them on. We typically use synthetic media. So we know the compounds that are on the media so we can control the nutrients that these microorganisms have. We also have minimal media, which has less nutrients, and some microorganisms are just fine on minimal media. They grow quite well. These are most of our laboratory rats that we use within the lab, meaning the ones that we use the most, like E. coli. But some bacteria need a little bit more nutrition associated with growing. So this is where we get into our complex media or enriched medias. So these enriched medias have additional growth factors for microorganisms to grow that wouldn't simply grow on minimal media. Some of these enriched medias will promote the growth of fastidious microorganisms. These are microorganisms, fastidious microorganisms, or organisms that don't typically grow on minimal media. So sometimes when we have difficult to grow microorganisms, we can use enriched media. Let's go ahead and talk about selective and differential media. So these are some great terms to know. Selective media allows for some microorganisms to grow while preventing other microorganisms to grow. Selective media can be compounded by using antibiotics in the media or chemicals that prevent the growth of microorganisms. So that allows the growth of some while preventing the growth of other. It's selecting for specific microorganisms to grow. On the other hand, we have differential media. Differential media allows for all organisms to grow, but we can determine the difference between the ones growing on the plate based off of unique metabolic changes. So in this example here, we see McConkey medium. It's actually selective and differential. So we can have both those components combined into one medium. McConkey medium is a really, really common lab agar that's used. Its selective component is going to be bile salts and crystal violet. So bile salts and crystal violet are going to prevent gram-positive microorganisms from growing, and also it allows for enteric microorganisms to grow. So it selects for those gram-negative enteric microorganism and cancels out the gram-positive organisms. So bile salts are similar to like what's in our stomach and our gastrointestinal tract. So we're growing these enteric organisms, microorganisms associated with the GI tract. And these are the gram-negative ones we're looking to isolate. Following that, the differential component of this media of McConkey agar is actually the lactose that's within it. So any bacteria that can ferment lactose is going to turn pink. Microorganisms that are consuming other nutrients in the media and not using lactose are going to just look like this off-white kind of color. So a kind of a cream color. So we could plate 2 different organisms on this plate and learn more about their metabolic properties. So whether they break down lactose, whether they're fermenting it, or whether they don't. Looking at this image on the right, that's a McConkey agar plate. Everything we see growing there, we know is gram-negative enteric bacteria because of the selective component that bile salts and crystal violet, again, select for microorganisms that are gastrointestinal and gram-negative in nature. Following that, we can differentiate between the two species of microorganisms there because we see one is fermenting lactose and one is not fermenting lactose. The one in pink is the one fermenting lactose. The off-white one is the one not fermenting lactose. Okay. Let's talk about living and dead cells. So if we wanted to plate microorganisms and see them grow, we can count live cells. So when we saw that we were doing those tenfold dilution schemes, we were looking at living cells and numerating the living cells. We can also look at the total number of cells in a sample, dead or alive. We have direct ways of doing this and indirect ways of doing this as well, like FACS. Let's go ahead and go over one way. So on the bottom left of the screen, we see a grid-like slide in which we can place microorganisms on. And the grid on the slide actually is what we can see when we put it underneath the microscope. And under the microscope, we can count per box, per square, how many microorganisms are in there, and then multiply that by our sample to get a good estimate of how many cells are in solution. Now, with this method, we may be looking at dead cells or living cells at this point. Looking at facts over here, we can look at turbidity within a solution as well, or we can see how we can use a laser to count cells. So we put our sample through, and the laser is actually counting fluorescence associated with cells as these fluorescent tags on the cells move through the instrument. Okay. Let's look at growth in itself. So microorganisms grow by a process called binary fission. Binary fission in bacterial cells is pretty unique. We go from one cell to two cell in a clonal division pathway. Clonal meaning the DNA is the same from parent to offspring. So from one cell to two cells, essentially the same DNA unless there's additional mutations. So this is a mitotic form of growth. Looking at binary fission itself, we can see that it can be symmetric or it can be asymmetric, as seen in these images here. Okay, let's look at growth patterns themselves. So once microorganisms start growing, let's say they're dividing every 20 minutes, we start hitting what we call exponential growth. So when the microorganisms have met the optimal conditions for growth, optimal pH, optimal temperature, optimal nutrients, optimal space to move in, they're able to go on exponential growth. So every 10 minutes, we go from one cell to two cells. Those two cells then go to 4 cells, 8 cells, 16, and that's our exponential growth. We can calculate the doubling time, or what we call generation time. Typically, that can be 20 minutes for microorganisms like E. coli, but it can be even less than that, or it can even be hundreds of years for some unique cell types. Okay, let's look at phases of growth. Here we see 4 different phases of growth: lag, log, stationary, and death phase. On the bottom x-axis, we're looking at time. And on the y-axis, we're looking at number of microorganisms in a logarithmic scale. The first part in purple are lag phase, which we see number 1 in the very left column. This is really the introduction of a microorganism to its environment, where it's testing out the pH, testing out the salinity, testing out the nutrients. If all of the optimal conditions are met, these microorganisms start activating cellular machinery to begin division of the cell. And that's where we hit the log phase next. This is where we actually start growing. We have that exponential growth, that straight line up into that we see here. So this is that doubling time over and over again from 2 to 4 to 8 to 16 to 32 and onward. Then at 3, we hit a stationary phase. We hit actually a plateau. What happens here is that microorganisms are assessing or reassessing their environment and realizing some of the abundant resources that they had before may be lacking at this point. So maybe they're running out of food, maybe there's an accumulation of waste, maybe there's no more space to grow and to divide. So reassessing their environment and realizing that there's a potential stress response going on here. So they stop dividing so that they don't cause additional stress to the population. And then, as these microorganisms stop dividing, they still live and they're still metabolically active in high numbers as we see. They're still consuming everything around them and still producing waste that at one point in stationary, we drop off into the death phase. So cells begin dying at an exponential rate. So we start dropping back down. Again, could be an accumulation of waste, could be lack of nutrients that cause them to die. Okay. If we want to prevent that death phase, we can actually grow microorganisms in something like a continuous culture. And it's actually analogous to what our body does. So if we're growing microorganisms in a fresh medium, as we see here, what we'd want to do, if we're growing microorganisms in fresh medium, we would be culturing them in a flask and constantly adding in fresh medium. So this means you're going to be getting more nutrients, new nutrients as they deplete the nutrients. And microorganisms produce waste. So just like humans, we have food in and food goes out as waste products that can build up and be toxic to them. And we can start filtering out the waste. So we remove waste. So by adding in nutrients, removing waste, these microorganisms continue to grow in this cultured flask that we see here. Okay, our body is very much like that. So our gastrointestinal tract is lined with microorganisms, and we're constantly bringing in fresh nutrients for them, fresh foods. We're not only feeding ourselves, but we're feeding the microorganisms within our body. And we're removing the waste products, not only that we generate, but also that the microorganisms generate as well in this process. So they benefit from this continuous culture aspect. Microorganisms, just like us, have essential nutritional needs. We see the essential elements they need, very similar to what we need as well. Any essential nutrient for a microorganism is a compound that microorganism cannot generate themselves, so they must obtain it from the environment around them. So whether that's through eating or through assimilation, they must be able to obtain this form of nutrients. Microorganisms also use growth factors, and growth factors are going to be different dependent on different species. This table shows quite a few different growth factors associated with different types of microorganisms. This is not something to necessarily memorize, but just to understand that microorganisms are extremely diverse and metabolically diverse too. So it makes sense that their needs of nutrients and growth factors are going to be quite diverse as well. Okay. Looking at culturing microorganism, we have to make sure that we give them the essential nutrients that they need. So not all microorganisms can be grown in the laboratory with the culture that we give them. Only about 5% of microorganisms that we know of can actually be grown in the laboratory, and it's due to a combination of factors. So some of those factors is that they have complex relationships, symbiotic relationships with others, so we can't grow them in pure culture. Or sometimes they're dependent on other complexes that we can't provide them. So we're unable to grow them. So this nutrients and environmental niches, it's going to be extremely important to allowing microorganisms to grow. We need to mimic their environment. Okay, let's talk about autotrophy and heterotrophy. So we may have heard these terms before. If we're looking at humans, we're known as heterotrophic organisms. We're looking at plants, those are autotrophic organisms. So the biggest difference here is that an autotroph is something that's going to be able to make their own carbon compounds like glucose. So the beginning of the food chain, that carbohydrate that we use. Heterotrophs, on the other hand, they don't make their own carbohydrates. They have to obtain their carbohydrates from other organisms. So already we're seeing an association with autotrophs and heterotrophs. Autotrophs are the ones supplying heterotrophs with the nutrients that they need. So autotrophs are like plants running photosynthesis that generate glucose. And then heterotrophs are like our cells, where within our mitochondria, we break down that glucose to make energy. Well, microorganisms are the same metabolically as us. They can be heterotrophic or autotrophic as well. Looking a little bit at the carbon cycle between auto and heterotrophic organisms, we can see here light absorption through phototrophy gives us glucose. That's our carbon-based molecule for food. We can also have something called lithotrophy. So in the absence of using sunlight to produce glucose, we can have lithotrophy. We can look at the compounds to make glucose, which can then pour into what we typically think about as cellular respiration or fermentation, where we're breaking down these compounds of glucose. Okay. Looking at phototrophic organisms, again, they're using light energy from the sun to generate glucose. Chemotrophs, on the other hand, they use a form of chemical energy, and this chemical energy can come in two different forms. It can be litho or organo. Litho meaning inorganic, organo meaning organic. So we have phototrophy and lithotrophy. Phototrophy and chemotrophy, which can then be broken down further to lithotrophy and organotrophs. Okay. Looking at phototrophy and chemotrophy, we can break it down further. Again, we said we had autotrophs and we had heterotrophs. Our energy sources, again, can be photo, light, or chemo meaning chemical. Then our electron source of this energy can be litho, inorganic, versus organic. Here's a couple different ways of looking at combining these terms. Not all of these terms would be necessarily found in nature, but it shows you how all of them can be combined together. We go from energy source to electron source to carbon source. So energy source will go photo for light. Electron source, we can go organo for organic. So it's photo-organo. Then is it autotrophic or heterotrophic? Let's say it's heterotrophic. It would be photo-organo heterotroph. So photo, we're getting light energy from the sun. The electron, we said electron source is organic, and heterotroph, it's going to get carbon from somewhere else. So just an example of combining these words. This is another way to do it as well. We could go chemo, litho, auto for a chemo, litho, autotroph. We go chemo, organo, hetero for a chemo, organo, heterotroph. Okay. Looking at some of the nutrients that plants need and microorganisms need, nitrogen is one of the main ones. And we actually know that plants can be dependent on microorganisms for nitrogen. So the nitrogen that we find in our atmosphere, it's about 79% of our atmosphere, is nitrogen gas. Well, nitrogen gas is not in an available form for plants to assimilate. So what has to be done is nitrogen fixation, and this occurs through bacteria. They convert this nitrogen gas in the atmosphere to ammonium, and then that can be used for biosynthesis within plants, as nitrogen is essential for plants. Looking at the nitrogen cycle, we can see here that nitrogen gas is removed from the air and converted to ammonia. From ammonia, we convert it to nitrate, and that's what we call nitrification. So from step 1 to 2, completing 2 is nitrification. Following that, we can go back into the gas form. Nitrogen is reduced to form a nitrogen gas. Okay, and that's our denitrification. So we see this cycle moving around from nitrogen fixers to nitrifiers to denitrifies. Okay, let's go ahead and look at different optimal conditions for microorganisms. As microorganisms are extremely diverse, we're going to see that what may be beneficial for one organism is not necessarily beneficial for another. So some of those conditions include temperature, pressure, osmotic balance, and pH, but there's a wide range of things to cover. When we talk about microorganisms living in these environments, we talk about normal physiological conditions. A lot of these are based on an anthropogenic view, meaning what we think is normal, what conditions work well for us. And anything outside of that is typically called an extremophile. So something that lives in a very cold environment or something that lives in a very acidic environment is noted to be an extremophile. We'll see some examples of these. Looking at the vocabulary associated with these terms here, we can look at temperature. We have hyperthermophile, thermophile, mesophile, which is considered normal, and then psychrophile. Looking at pH, we have alkophile, neutrophile, acidophile, neutrophile being the normal. Osmolarity, so how much salt in a concentration can a microorganism handle? Well, a halophile is a microorganism that loves high salt concentrations. When we look at oxygen, we're going to look at strict anaerobes, facultative anaerobes, microaerophiles, and strict anaerobes. We're going to look at all ends of the spectrum when it comes to oxygen, whether microbes want to be around oxygen or don't care. And then we get into pressure. We see barophile and barotolerant. So this is a great chart for organizing the terms. We're going to go into more detail on all of the following slides here. One to start with, let's go ahead and look at temperature. So mesophiles is our average temperature. This is in Celsius, so about 37 degrees here. That's again based off of our anthropogenic view. And here we see the growth of the microorganisms. So once it grow around 15 degrees Celsius are considered psychrophiles. Then we move up to thermophiles and hyperthermophiles at the very hottest. Thermophiles themselves are typically around 55 degrees Celsius. We could maybe find this in Yosemite National Park, where they have these geysers, this warm water. We're finding microorganisms that live in these warmer conditions, these thermophilic microorganisms. Following that, we have hyperthermophiles. These are the microorganisms that are growing in temperatures that are extremely warm. If we look at the bottom of the ocean, we have what they call these black smokers, and the temperature down here is around 100 degrees Celsius. But because the pressure is so high, we don't necessarily see boiling, but 100 degrees Celsius here would be boiling water. So we have microorganisms that can withstand these extreme temperatures, and they're completely viable here. Those are our hyperthermophiles at the bottom of the ocean. Looking at psychrophiles, on the other hand, psychrophiles are microorganisms that grow at low temperatures around -10 degrees Celsius. For reference, water freezes at 0 degrees Celsius, so -10 is even colder. So this is something we could find in our freezer, a psychrophile. Then we have our mesophiles. That's going to be our normal standard temperature, about 37 degrees or so, or 20 degrees Celsius, anywhere around there. These are our lab rat microorganisms that we use because they grow in a generally well-conditioned environment that we live in as well. Okay. We also have microorganisms that can be considered extreme based on the environment that they're in. So we have barophiles and pisophiles. These are microorganisms found very, very low at the bottom of the ocean, like the Mariana Trench, because they're withstanding extreme degrees of pressure. And these cells are not imploding. These cells are living and thriving and dividing at these environments. We also have hyperbaric environments where we have very low pressure conditions, so up in the atmosphere. And we know microbes that can live in these low pressure environments as well. Let's look at salt concentration. Here we see a salt bed. Well, many microorganisms cannot grow around high concentrations of salt, but some microorganisms are what we call halophiles, and they live at an elevated degree of salt, a higher concentration of salt. When salt is around cells, it changes osmotic regulation. So when we talk about the osmolarity of these solute molecules, we're going to look at whether these microorganisms can tolerate an environment, maybe that's hyper around them, that has more salt concentration. Let's go ahead and check out pH. So we have our neutral pH at 7. We would call microorganisms living at this pH neutrophiles. Acidophiles getting closer to 0, so 0 through 5 here. Then alkaphiles are basic ones, 9 all the way up to 14. pH is actually the negative log of the hydrogen ion. And what's great about this image here, it's actually showing us the relationship of hydroxide to hydronium here, which is our H3O versus OH. When we know that as we move into acidophiles or even just hydrogen ion, as we move up, we're going to increase the amount. And over here, we increase the amount of the hydroxide ion. Okay. Looking at pH in the environment, neutrophiles are the microorganisms that live anywhere from 5 to 8 on the scale. So that's kind of our average area. 5 to 8 around here would be a neutrophile. Acidophile, on the other hand, are microorganisms that live at those very low pHs. And alkaphiles are the ones that live at those very high pHs. Unique situation that you may have heard of before is with flamingos and pigmentation. Flamingos live in very alkaline environments. This alkaline environment allows for the growth of algae and other microorganisms that produce these carotenoids that are actually pigmented, kind of an orange pigmentation. And what happens is that brine shrimp eat these microorganisms and this microalgae, and then the flamingos end up actually eating these brine shrimp that have eaten the microalgae itself. And when it consumes the microalgae and when it consumes the carotenoids that were previously concentrated by the brine shrimp, it starts concentrating these pigments, these molecules that contain color within the feathers of the flamingo. So that's why we see this pink color. Flamingos don't initially start out pink. They turn pink because of this accumulation of pigments, essentially from microorganisms. Okay, let's talk about oxygen requirements with microorganisms. This is a big one that we have quite a few different terms for. So we can start with aerobic and anaerobic. Aerobic deals with oxygen. So we have strict anaerobes. These are microorganisms that need oxygen for their metabolism. These microorganisms are also able to deal with reactive oxygen species, what we call ROS, which can be actually damaging to the cell, but they have the ability to detoxify ROS. So these microorganisms are living in an environment with a surplus of oxygen. Strict anaerobes, on the other hand, are the exact opposite. They don't want oxygen around them, and ROS, that reactive oxygen species, can build up in a high concentration that ends up being poisonous to them. And in between aerobic and anaerobic, we have a couple other terms to introduce you to. Here we have aerotolerant. So these are anaerobic microorganisms, but they're not too affected by ROS, reactive oxygen species, and they normally lack catalase. So we have different metabolic reactions occurring here. Our facultative anaerobes, on the other hand, can switch back and forth between aerobic and anaerobic. When they switch back and forth, that means that they can use oxygen, they can survive in oxygen, or they can survive without oxygen. So they're facultative, they're moving between. Then we have microaerophiles found in this image, circled in yellow. Microaerophiles are kind of borderline. So when we see them, they're actually aerobic microorganisms, meaning that they can live in environments with oxygen, but they prefer these environments with lower concentrations of oxygen. That's why when we see this test tube here, we see aerobe, microaerophile, anaerobe. They're in the microaerophile area because they need just a little bit less oxygen than their other aerobic counterparts. Okay, aerobes versus anaerobes. Here's just another chart of information of microorganisms that fall into these categories that you can take a look at later. Let's talk about how we grow some microorganisms in the laboratory if they don't want oxygen around them. So if we have our anaerobic organisms, we need to either grow them in an anaerobic jar, as seen on the right. This gas pack actually removes all the oxygen and generates CO2, so we don't have oxygen preventing the growth of microorganisms. Then we also have anaerobic chambers. So this is an entire enclosed chamber here, and where the atmosphere is actually replaced either with nitrogen or carbon dioxide gas. So there's no oxygen getting into this anaerobic chamber. Let's talk about microorganisms in nature. So in nature, microorganisms exist in biofilms. So we can have hundreds of different species of microorganisms living together. Most of them are communicating and working together well. Some of them are fighting a bit, but this image of biofilms is actually maybe something we can all relate to. If we stop for a minute and lick our teeth, you can feel a grimy buildup if you haven't just brushed your teeth relatively recently. And what this is microorganisms sticking to the surface of our teeth. Let's go ahead and see how biofilms are produced on our teeth. So biofilms are produced with the process of quorum sensing, and this is how microorganisms actually communicate with one another. So they're actually talking to one another. So now that we've felt the bacteria in our teeth, we can think about the fact that they're actually talking to one another while they're on our teeth. Let's go ahead and go over the stages of biofilm production. We have step one. This is where microorganisms swim to a surface and they go ahead and start making a monolayer, a single layer. So they swim and start adhering. Once they get to that surface, we can begin step two, which is where they start a twitching motility, which is where they kind of move back and forth on this solid surface. So they move a little bit and they try to accumulate with one another. So now we're no longer a monolayer, kind of starting to be a grouping of cells. And once they get together, they start talking to one another. They start coordinating and talking through this twitching motility of where one is so that they can round up one another into these groups of cells, into this biofilm. Then once they're close enough, they start secreting this compound called EPS, extra polymeric substance. And it's like this sticky goop around them. And that's what we see in green here. So this is kind of the very beginnings of biofilms. And here we see mature biofilm towers. So cells are communicating and they're building up in pillars in these large communities where we can have hundreds of different species of microorganisms working together and communicating together. Biofilm towers, like you see here, eventually get dissolved. Let's say we brush our teeth and we knock off the biofilm or just naturally knock it off. Microorganisms will be released again just to form another biofilm. Okay. For one of the last topics in this lecture, we're going to go ahead and talk about endospores. Endospores are really unique because it's essentially a dormant phase of a cell, meaning they're not metabolically active. Sometimes we can compare this to bears that are hibernating over the winter. When bears hibernate over the winter, they slow down their metabolism. Endospores, on the other hand, they survive a long duration of time by actually preventing their metabolism from occurring at all. So that means they're not making any energy, they're not consuming any energy, and they're basically closing down into the spore format so they can survive in a longer time. Cells move into this endospore format when the environment around them is not desirable. So maybe not enough food, not the right pH, not the right temperature. They go into this form so that they can preserve their genetic material, their DNA for later, and be able to reemerge at another time. Many of the cells that are endospores that we see are like Bacillus and Clostridium species. These can all form endospores. Okay. Endospores can survive in nature for hundreds of years, thousands of years, and some suspect millions of years as well. So if we're looking at permafrost or in glaciers, we have endospores that have been frozen for millions of years that we're actually still able to get vegetative cells out of. Quite interesting. Let's go over the process of forming an endospore. So step one, we actually get this septum. So this is the beginning of separation between two cells, and our DNA begins replicating. So we go into a copy of our DNA here. So replication, following that, septum separation continues, and we keep separating out our DNA more and more. It's being pumped through the septum so that we have the equal concentration of DNA between both cells. This looks like a typical binary fission example that we're seeing here. Following that, the septum completely grows and separates the spores. We have this spore here. And then what we see is actually the mother cell actually engulfs the spore, and that gives it a second membrane. So now we have an additional membrane surrounding this genetic material. What we see here is that this other genetic material that we had before will actually be of no use any longer. So it will be broken up and disassembled, and we'll start building a cortex around this DNA because we really only need to preserve part of the genetic makeup or one cell's worth of genetic makeup, not two. Following that, we have a cortex, a very, very hard coating onto the cell, and we see we're still dissolving the DNA. So here's our spore coat, as we see here, then the mother cell can release the spore, as seen here. What can happen is what we call germination. We go from a spore, this completely dormant phase, the metabolically inert cell, back to a metabolically active cell. So we can germinate back. And here we see we can become a normal cell and go into what we call vegetative growth. We can go ahead and run binary fission again. Okay, we've now reached the section of this where we start answering questions. Question one says, you are interested in determining the total number of viable cells in a bacterial culture. Which method of counting would you choose for the most accurate results? Again, this is the time where you pause the video and think about it. And then I go on to the answer. So this would be a spread plate dilution. The reason why we would choose a spread plate dilution here is because the microorganisms are living. They need to grow in order to be numerated here. So they're living. Other methods that we're looking at here don't necessarily tell us the difference between living cells and dead cells. And the key in the question was that we wanted viable cells. Question number two, a culture of bacteria is growing exponentially under ideal conditions. Its generation time is 20 minutes. The culture starts with 2 bacteria. How many bacteria will you find after 1 hour of growth? So again, you can think about this and start doing simple math. You may or may not need a calculator for this one. We'll go ahead and move on to the answer of this one. So the answer is 16 cells. So remember, we start with 2 cells, and we're going for a total of 1 hour with 20-minute cycles. So from 2 cells after 20 minutes, we go to 4 cells. And then from 4 cells, after another 20 minutes, getting us to 40 minutes, we're at 8 cells. And then we go another 20 minutes, we go 8 plus 8 gives us 16. And that's our entire 60 minutes or 1 hour of growth. So 16 cells from 2 cells to 4 cells to 8 cells to 16 cells. Okay, question number 3. How would you classify a bacteria that uses CO2 as a carbon source and gains energy from hydrogen sulfide in an organic compound? So go ahead and review the questions or answers, and we'll go ahead and move on. This would be an autotroph and lithotrope. So if we look at the keys in the question itself, so the bacteria uses carbon dioxide as a carbon source and gains its energy from hydrogen sulfide, an inorganic compound here. We know that it's going to be autotrophic because it's using CO2 to make carbon. And it's a lithotrope because it's using an inorganic compound for that. So it's autotrophic and lithotrophic at the same time. Let's move on to question 4. One likely adaptation from microbes that live in psychrophilic environments is that. Okay, moving to the answer for question 4 is that their membranes are more fluid than mesophilic bacteria. So if we go back to psychrophilic environments, these are those extremely cold environments. In extremely cold environments, we have less movement. So as we heat things up, molecules move around faster. So what needs to happen in these cold environments is that the membranes still need to be fluid, right? So we have to ensure that the membranes maintain that fluidity. So they would evolve to change their chain structure, their lipid membranes, in order to be more fluid. So maybe they have shorter chains, or maybe we have more unsaturated fatty acids that make them more fluid. Okay, looking at question 5, how would you classify a bacterial species that grows only in the highlighted area of the culture tube? So the highlighted area is low oxygen here. Okay, the answer for question 5 would be a microaerophile because it survives in low oxygen. Okay. So these are our section objectives. Again, at this point of time, this is where you would stop, do a study session off of these. You'd go back through these topics and start filling out what you know. Essentially, after each lecture, you're building your own study guide, whether you're working with other students or whether you're just working on your own. Okay, that concludes this lecture.